Assalamu alaikum. I'm Khadija Askar from 7th semester. The topic of my presentation is phenotyping and plant breeding, overcoming the barriers. So firstly we will define plant breeding. All we know that about plant breeding is a science and art in which we develop the varieties with desirable traits, higher yield or resistance to biotic or abiotic stress. So now we will talk about phenotype. Phenotype is the science basically in which we observe that data by physically appearance, biochemically, or in short, we see the all characters of plants, like their color, their height, uh, all that things that we observe physically. So in short, we will discuss the barriers that we face in phenotyping. So barrier one is basically limited seed supply in early segregating generations. सो इस पॉइंट में जो अर्ली सेग्रीगेटिंग जनरेशन्स है ना, आफ्टर क्रॉस हैव लिमिटेड सीड्स ईच रिप्रेजेंटिंग अ यूनिक जीनोटाइप. This limitation is basically often forces breeders to rely on visual selections, breeder eyes, until enough homogeneous seed is produced. Another step similarly like limitations जो occur होती हैं when we are working with crop with relatives, CWR or gene bank materials like genotype data, something like that, using individual plants in honeycomb selection designs, HSD in shortly, or remove seed supply constraints and allow early multi-locations and evaluations. This is all about first barrier, and now we will talk about second barrier. Second barrier is effects of interplant, ignore my handwriting please, competition. In second barrier, interplant competition is basically the mask in which we perform true plant performance or reducing selection efficiency. ओवरकमिंग जो है इस पे हम लोगों ने करनी, मतलब इस पे हम लोग किस तरह से कंट्रोल कर सकते हैं, वो इस तरह से लाइक पार्टीशन क्रॉप पोटेंशियल, ठीक है, मतलब क्रॉप का पोटेंशियल स्ट्रॉन्ग हो, ठीक है. सेकेंड थिंग, डेवलप होल प्लांट एंड सिबलिंग लाइन फॉर एक्यूरेट प्रिडिक्शंस. Then the third point for overcoming ranking under ultra wide spacing corresponds close to commercial densities, removing the need of optimal planting density. Next goal. You all know when we talk about problems, then we discuss overcome, then we discuss goals. So goal are develop density neutral or density independent varieties, crucial under climate change, drought prone conditions. Dense planting hinders field phenotyping, or especially for canopy crop roots. Now we will talk about the third barrier, and third barrier is effects of soil, like how soil affect the phenotypic data and all. Soil